So it's my pleasure today to introduce Dr. Dennis Whigham. Comes to us from Smithsonian Environmental Research Center, the same place that Tuck Hines comes from last week. Dennis got his PhD at the University of North Carolina at Chapel Hill. He was a professor for about 10 years at the University of Utrecht in the Netherlands, and he served as the deputy director of CERT for the past 5 years. He tells me that his real research interest can be described as plants and plant biology, but he's interested in all manner of plant biology, including plant ecology. He's worked on a wide range of things and continues to work on a very wide range of projects. They include work with Phragmites australis biology and its ecological impacts, and trying to determine what the basis of its establishment is, whether it's dispersal, for example, from vegetative fragments or from seed. He is very interested in, works on rare orchids. He's interested in the consequences of global climate change on mangrove forests, and broadly speaking is interested in wetlands ecology. He likes to use an interdisciplinary team approach to research and work with a variety of people to bring different skills to bear on projects. One of his activities recently has been trying to establish and launch the North American Orchid Conservation Center. He's published more than 200 papers, and just to give you a sense of some of his work, recent paper that we had talked about was with a variety of folks in his lab collaborators, but it's limitations on orchid recruitment, not a simple picture. That was published in Molecular Ecology, and details fungal associations with orchids as a limitation of their recruitment. So today, Dennis is going to talk to us about some of his work in Alaska, and so I'll let you take it away. Thanks, Dennis.
Thanks. Nice to be here to watch you all eat your lunch. I was telling Matt, my wife and I started on a thing last week on low glycemic, so if I just collapse in the middle of this, you know, go over and get a Coke or something. It's a pleasure to be here in your new building. The last time I was here, Matt reminded me, it was either a hole in the ground or something emerging from a hole in the ground. We're in the middle of building a new lab at our place too that has many of the features of your labs, which is shared space, so it's nice to see that today. So yeah, I'm going to take us to a place that's kind of cold right now. This is a photograph taken from Homer, Alaska, across what's called Kachemak Bay to the Kenai Fjords National Park that you see there, a bunch of glaciers up there. Kachemak Bay is a big body of water, but it's just a small part of Cook Inlet. So I'll show you a map of that in a minute. Whoops, already starting off well. So there's the title. This is really a talk about landscape ecology, which has been an interest of mine for many years and continues to prick my interest whenever I have an opportunity to look at it. So what I'm going to tell you today about is a sort of a story that's still unfolding, but we think we're fairly far down the road to understand how headwater streams support juvenile salmon, and I'll explain that in a little bit of detail. And as a wetland ecologist, I'm interested in how wetlands fit into that picture, and that's where the Bluejoint comes in. It's a dominant wetland species there. And then the other that sort of kicks the biogeochemistry is alder, which you may already know is a plant that has bacteria associated with its roots that fix nitrogen. So alder is going to play a key role in the nutrient dynamics of these systems. And as Matt also mentioned, this research and others that I do involve usually a cast of characters. In this case, people from the Estuarine Reserve site in Homer. It's one of the National Estuarine Reserve Program sites at Kachemak Bay. Ryan King is a former postdoc of mine who's now a professor at Baylor University. He's doing the stream ecology. And Mark Rains is a hydrogeoecologist. He's at the University of South Florida. He's going to he does the water sorts of things that we're interested in. And then Cooey Walker is sort of she's sort of, I would call her sort of the head behind this. She's an incredible person, actually, and she's the one who sort of cuts through everything and helps us get funding and communicate what we want to do to the public. And so that's sort of the main cast of characters that are involved. And Steve Baird he's a GIS guy. I thought before I would start, not knowing who I was going to talk to today, that I'd give you some idea of some of the other projects that I'm involved in. Matt already mentioned this one of Phragmites, which is a NOAA-funded project that has a whole series of institutions involved. The piece that we have is about a third of the project that focuses on the invasion of this Phragmites that you see here on the shoreline, which is just going like crazy within the Chesapeake Bay, and we're trying to understand what the ecology is behind that, but more importantly, how human activities are involved in it and the consequences of that, hopefully leading eventually to something that deals with control. This is the most endangered plant in North America. It's a little orchid about this high that has populations from Georgia up to Maine. It's in big trouble in the southeast, and we have a grant from the Park Service to try to understand its ecology well enough to even begin to make an educated guess about what we can do to keep this plant from going extinct. We're doing this work at an Army base south of here, at a national park south of here, and we're involved with some other folks in various ways in the project. And this is a small project that involves my collaborators from the Netherlands. This is Joost Verhoeven, who has been a long-standing friend of mine. We've done a lot of work together over the years. And this work takes place in Florida. It's funded from a small program that the Smithsonian has. And the idea here is what happens as the climate warms and as mangroves move north and they invade into salt marshes. And our particular interest here is on the microbial community. Does the microbial community change because there's chemistry associated with mangroves that potentially will influence the microbial community, and then how does that affect ecosystem processes? And then where I'm spending actually most of my time now is it's kind of quasi-science. It's a spin-off from my long-term interest in orchids, the ecology of orchids, and a new initiative at the Smithsonian in collaboration with the U.S. Botanic Garden. We're trying to set up a program that, long after I'm out of the business, will assure that every of the 250 orchids in North America, they can survive. We can assure their survival in one way or another. I won't go into the details of that, but it's going to take a lot of money, a lot of interaction, but it's off the ground and sort of going. But this goes back to a project that we had some years ago that was funded by EPA. And our part of that project was that we looked at watersheds all around the Chesapeake Bay that were dominated by different things. Our sort of the control would be something that was dominated mostly by forest. That's sort of what it used to be like. And then all the way to things that were highly developed. And the idea behind this project was to ask, what are the consequences of human activities on the land in aquatic resources, whether they're in streams or in estuaries? And there was a large team involved in this project. And this sort of is the take-home message. We found that there were many indicators in streams and in estuaries, their water quality, animals, wetland plants, that were responding to what humans did on the land. And almost in every case, they were most strongly related to the amount of development that occurred on the watershed that surrounded the body of water. And the other thing that wasn't surprising necessarily, but what was really surprising was the shape of every one of those response curves. And that was that things seemed to be okay, and then once you reached a certain point, you went through a threshold, and then things really changed rapidly. And in this case, this red line is for Phragmites, the abundance of Phragmites in brackish wetlands. And you can see that by the time you get to about 20% of the landscape being developed, there was a strong response to Phragmites. And when we looked at that in more detail, it was not only that the land was developed, but how close that development was to the water. The closer you got to the water, toward the shoreline, the stronger the response, which is sort of summarized there. There was a threshold for almost all of these variables. So this is Homer, Alaska, and my daughter lives there, and I have grandchildren there. So I'd been going there for some time, and I had already developed a relationship with the people at the Estuarine Reserve. So after doing the EPA project, I said, well, wouldn't it be fantastic to go to some place where we haven't screwed it up? Where obviously humans are there, but we haven't dominated the landscape to the point where we really influenced these aquatic resources. So let's go look for such a place. Let's try to understand how that landscape fits together so that we can ultimately provide management with the right kind of information to hopefully avoid the problems that we have down here in the lower 48. So here's Homer, Alaska. This is where we're based out of on this project. The picture that I showed you at the very beginning was standing here, shooting across here. Anchorage is located there. You kind of drive down from Anchorage about 4.5 hours to Homer. These are called the Kenai Lowlands, and this is the study area that we work in. A few pictures to kind of set the mood. This is driving down from Anchorage, a little past Turning an Arm, just on the way down almost toward Seward. This is an autumn photo, which is like third week in August. And here's a picture taken in early September. This is on the kind of when you drive out of Homer as far as you can go on the road up to the head of Kachemak Bay and look across. That's what you see. Beautiful landscape. One of our study sites on the Ninilchik River. So we're out here studying whatever we study and looking across at this lovely volcano. There are 5 of them that you can see as you come down from Anchorage on a clear day. You see all 5 of them. Sometimes when you're there, the scenery is a little bit different. There are massive wetlands up there. I'm going to come back to that point in a minute. These are brackish or saline wetlands. There are humans out there. There are other things that go out there every once in a while. This area is known for its fisheries resources. Homer is a small town, but it's sort of up there on the scale of fish landings for the U.S. And this photo was taken probably about 11:30 at night in the middle of the summer in the Homer Harbor. It's called the halibut capital of the world. So lots of people make journeys there just for catching halibut. I try to catch some. Well, you don't have to try. You just put the thing over the boat and they come up. It's like dragging up a log off the bottom of the ocean. There are other things up in Homer. Here's me working with my son-in-law on their oyster farms. We're washing these oyster nets. They have the most fantastic tasting oysters in the world. They're not native there, though. They have to be brought in. So my son-in-law also, he's a commercial fisherman in July, and this is one of his catches on a given day of sockeye salmon. So the one thing that they all do in Alaska is they have a common language. It's called salmon. So anything that affects that resource, you can get their attention. So as I mentioned, we started working with the folks at Kachemak Bay Reserve, and we initially had funding from EPA to get this project off the ground. And since then, we've had funding from the Alaska Salmon Fund, which is it's money Congress gives to states to study Alaska, and they partition it out among the different states. So we fortunately were able to get one. The people who work with me on this project, here's Ryan King, who's an avid fisherman, and you can see he knows how to do it. Here he's got a Chinook salmon in the Anchor River, one of the systems that we study. Here's Cooey Walker, who's our head honcho up there at the Estuary Reserve, and Steve. This is a lovely summer day on a tidal wetland. Picture of Steve on a day when he and I were going out to sample one of our wetland sites. And we were walking in, and I saw this, and I asked him what it was, and he said, well, that's where a grizzly sort of works its arms muscles. It just reaches up and strips the bark off the tree. So there are other things up there. And here's Mark Raines, who is weighing a little, looks like a Dolly Varden over there. So back to the map, and here's where the project focus is. So if you don't know what first order streams are, they're also called headwater streams, so that when you walk from high ground to low ground, eventually you come to a stream. That's the first order stream. So that was the focus of this project for a couple of reasons. One is Alaska has maps of what they call salmon streams. And you get these maps, and the map expresses basically where the adult salmon go. They swim up, they can only go so far, they stop, lay their eggs, die, and that's it. Well, it turns out that there was some knowledge that a couple of the salmon species, after the eggs hatched, the young went upstream rather than running out to the ocean right away. So we knew that, but no one had really quantified it. So our interest from this Chesapeake Bay project was, if we have the land interacting with water, the best place to go look for that interaction is right at those headwater streams. So we focused on headwater streams because of the landscape interactions, but also, and that's where human activity would occur, but also because we knew there were juvenile salmon there. We just didn't know which ones and how abundant they were and all that sort of thing. And our first thinking was that we would want to understand the relationships between landscape geomorphology, the physical features of the landscape, and what occurs in the streams and in the wetlands associated with those streams, with a focus on young salmon. So these were our original project goals, looking for landscape features that in some way would tell us something about the water chemistry, the hydrology, the habitat characteristics of the streams, the wetlands associated with the streams, the invertebrates in the streams, and then finally the fish, basically looking at food webs. And our hypothesis was that the geomorphology would be the primary driver. So that if a headwater stream was flowing out of a vast peatland, very low nutrient peatland, that would be different than a stream that was flowing out of a terminal moraine of a glacier, because the chemistry would be different. So our initial assumption was that we would see differences among headwater streams based on their origin. What was the geomorphology of the place from which they came. So that was what our first hypothesis was. There's a management side to this, which I'll come to at the very end, and that is that we benefited by having had available to us a document, but also web-based, in which all of the geomorphic features of the Lower Kenai had been mapped. We had that information. We also had maps of all the wetland plant communities in the Lower Kenai. So we were coming from a point of, having information you rarely ever have for a place like this. So one of the ultimate goals for this, other than those of us who want to know the science, is that how can the knowledge that we gain be used to benefit people who are interested in managing resources in that part of the state, basically. So I'll come back to that at the very end. So we spent the first summer doing a lot of backpacking into these sites, sometimes hours of backpacking. It's great for the being in shape. Sometimes we couldn't get very close, so we would get in four-wheelers and go off to wherever we were going to work. We didn't carry guns with us, but we did have bear spray all the time. Sometimes the car would not get to where we wanted to go because the bridge had not been maintained, and that kind of situation occurred. As I mentioned, we had this classification of wetlands and the geomorphology available to us, where they knew what plant communities were there and they knew what the landforms were. So that was an important component of what we did. So there are 10 geomorphic types in the Kenai lowlands. In terms of the aerial extent of them, there are 5 of them that are important that we focused on at the very beginning. Kettle wetlands. These are basically large peatlands that are associated with terminal moraines of glaciers. They're basically just great big old peat bogs. These drainage ways are very large features in the landscape where historically, when all the sediment came down from the glaciers, they basically filled up valleys, and you just had these very long linear features that are called drainage ways. We also had lake beds. These are typically places where a big block of ice had been deposited during the Pleistocene, and it melted away, and you had a lake, and it filled in traditional successional processes. Discharge slopes are places where you have probably a moraine, some kind of geologic feature, and groundwater is emerging, and then where the groundwater emerges at the base of that feature, you have a big old wetland. So that's another type of wetland that was very common there. And then one that doesn't quite fit in from a geomorphology context is what Gratz and all who did this project called riparian wetlands. And so these are streams basically that have definite channels with bank features and all that sort of thing. That's where our headwater streams fit into that sort of classification. So to make the point that wetlands are important, as the wetland scientist, everything blue there is wetland in the Lower Kenai. So for the area that they mapped, over 40% of it is mapped as wetlands. So wetlands, you know, have to fit into this in some significant way. The other thing I mentioned is about the first-order streams. One characteristic of first-order streams is that on any water, almost any watershed, they account for more of the linear distance of the stream system than any other stream category. So that's where most of the water is, not most of the water, but most of the stream distances, the linear distances. So this is an example of one of the watersheds that we worked in, the Storiski, where that watershed had over 50% of the total area was in these tiny headwater streams. So the message here is that that's a really abundant feature in the landscape. And here's a map showing those 5 geomorphic classes that I showed you a minute ago. You see a couple of things. Obviously, they're not distributed uniformly, that you have more discharge slopes up here close to the mountains, and you have more of the lake beds sort of associated with the western part where it dumps into Cook Inlet is over here. These little dots are our study sites. Homer, again, is down here. And this is the same map, basically on a different background to show the 5 watersheds that we worked in. So what we wanted to do was distribute our sampling across as many of the watersheds as we could within the Lower Kenai system. And every one of these, the Ninilchik, the Deep, Storiski, North, and South Anchor, these are all salmon, they all have salmon runs. And then the other thing I want to point out here is the blue maps are the ones that the state has mapped as salmon streams. The red ones are ones that, just based on doing GIS modeling, predicts a stream is there. And these are all headwater streams, first-order streams. And you can see there's a lot of red in that picture too. So if you're asking where are the salmon in the Lower Kenai, based on the blue maps, you might be missing part of the story. A few photos of what the streams look like. This is a headwater stream. I believe this is on the Ninilchik. This is very typical of a summer photograph in this kind of setting where the stream is incised in the landscape. Wetland vegetation on either side. The streams tend to be small and very shallow. Here are headwater streams associated with 2 of those geomorphic classes, a drainage way and a riparian system. They don't look that different to you. But notice again, lots of wetland vegetation immediately adjacent to the streams. Here's a stream. You might ask where. The way we would find some of these, when we would pick where we were going to work, this was all done randomly. We basically picked points using GIS, and then we would look at all the information we have and ask whether that was a good place to go to, and then we put on the backpacks and we would go. And a few places we would get there and there wasn't a stream, or we would get this kind of a stream. And usually when you were in this setting, you heard the stream. You didn't see the stream until you went down the gradient. The stream's there, so to sample it, what we had to do is crawl down a hole to go down to get the water samples and take the other measurements that we were taking. So when we would get to a site, put out the equipment, we usually would have, this is the early part of the project, we'd have one team doing stream morphology, collecting samples for water chemistry, things like that. Another group would be doing stream invertebrates, and another group would be electroshocking for fish. So we had people kind of doing all kinds of different things. Now I'll start showing you some of the data. This is after the first year of sampling. So all of the points on this plot are sites that we visited. And the arrows basically represent the metrics that we measured at those sites that allowed us to decide sort of what was going on there. And when you see these things are fairly continuous, but definitely there's a group over here and there's a group over here. And it turns out that all of the sites over on the right-hand side are high gradient, they're headwater streams in high gradient watersheds dominated by riparian forest and located sort of away from Cook Inlet on the interior side of the Kenai lowlands. So that was one group. So these are basically now, from this point on, you can think about what I'll say is that these are streams that are on a slope. They have a gradient. Are streams that are associated with very low gradient systems. The water's moving much more slowly, and they tend to be much, the wetlands tend to be much bigger. The substrate is peaty. whereas the substrate over here is gravelly and cobbly, so just very different stream sort of settings. So it turned out that geomorphology, you know, the source of the stream, was not important at all. The only thing that was important was features related to the stream gradient, everything that sort of subsequent and follows with that. Now that has consequences to ecology. So for example, if you take those sites, and basically when you get to these headwater systems that are on slopes, that's what we're going to look at now. So if you increase the slope, how do certain characteristics of those streams change? So for example, as you go up to the higher gradient streams, the amount of oxygen in the water changes. Well, that's obviously going to influence the critters that are in the stream. The saturated measurement of DO goes up. Stream temperature goes down. The slower it's moving, warmer. Okay, a lot of these things are just intuitive. pH goes up. The more you're up on these rocky, pebbly places, the more you're in contact with geology. So pH goes up. Dissolved organic carbon goes down because the PD systems, you got a lot of DOC and all sorts of forms of carbon there, so that goes down. But when you're back up on the landscape again, the inorganic nutrients will go up because you're potentially closer to sources. So dissolved inorganic goes up, ammonia goes down. The slow-moving systems, there's more, it's not anaerobic, but basically you have an environment that supports ammonia more. Phosphate goes up, the closer you go up, further up the gradient, and dissolved inorganic, the ratio of that dissolved and total nitrogen goes up too. This is published in a paper in Wetlands last year. In terms of the fish, here is an image of doing the electroshocking, and we published a paper in Freshwater Science on that last year, mostly on the geomorphology and the macroinvertebrates and the juvenile fish. And this is sort of summarizing that. These slower, lower gradient systems support larger fish. Again, it's fairly intuitive. These are coho salmon greater than 10 centimeters. The intermediate sites have larger numbers of small coho, so they're partitioning it based on size of the individuals. And then Dolly Varden all of these fish are everywhere, but the Dolly Varden is one that is in the very high gradient streams. So the fish are sensing these differences in stream characteristics based primarily on wetness and slope. Now this shows some of the characteristics of the fish related to some of these stream-related features. So the coho salmon that are greater than 10 centimeters, as the slope goes up, their abundance goes down. The small coho are sort of in the middle here, and the Dolly Varden that are larger are sort of in the middle, but displaced a little bit up from the coho. And then when you get to the smallest Dolly Varden, they're further up in these streams that are on a high slope. How does that relate to characteristics of the stream? As stream velocity goes up, you don't find the large fish, and at the other end of it, as stream velocity goes up, that's where you tend to find all of these small Dolly Varden, and the other two sizes are sort of intermediate to that. So stream velocity is important. The amount of oxygen, that passes the gills of the fish, you see the same sort of thing. The steeper, the more dissolved oxygen you have, basically, if you're a smaller fish, you're more sensitive to these sort of physical changes, and you just need lots of oxygen. So you're going to be in a place where that's not a problem for you. And they're also feeding all over the place, so the substrate becomes important. And these small fish are basically feeding in places where there's stones and pebbles and cobbles. And when you get up to the amount of very fine substrate that you have, you don't find most of these fish. That's where the larger fish are. Stream invertebrates, same sort of pattern. They weren't related to the geomorphology. What they were related to was the topography again. So over here, these high gradient streams had cleaner type, riffle type, mayflies, stoneflies, blackflies, and when you got over here to the slower moving streams, low gradient streams, that's where you had segmented worms, clams, caddisflies, other sorts of microcrustaceans. So the invertebrates were partitioning the landscape also based on these physical characteristics of the landscape. Now we have to bring in hydrology because this is all based on water. So here's Jason Bellino, who did his master's on this project. What he's doing is putting in a well so that we can measure groundwater. And one of the questions we had after the first year was we had all these fish all over the place. I don't know if anybody's been to Alaska at this time of the year. It's cold up there, and things freeze. So one of the big questions was, where are these fish going when it's frozen? And these tiny streams are no deeper than that. So we had to figure out whether or not the fish were actually there year-round. That was one of the things we had to do after the first year. And part of that was understanding the hydrology of the system, because if the fish are there all year long, winter long, that means they're not freezing solid. That means there has to be water. So we had to understand that sort of thing. So we put in a lot of devices. Here's Mark Raines. He's not grimacing because somebody's biting at him. He's grimacing because the water is about 1 degree centigrade. And what he's doing is putting a temperature tidbit down on the bottom of one of these peak-dominated systems. And so we put tidbits there. We put barometric pressure devices, and we just let them be there all winter. Came back the next spring to see what we could find. And here's an example. So this is a higher gradient system. And if you look at the groundwater, this would be freezing. What you see is that the groundwater is always above freezing. So it's always moving toward the stream, even though the air temperatures are way below zero. And then for the surface water, it's almost always above freezing, but there are a few periods when everything may freeze. But other than those few periods, it's always above. And then when the temperature starts changing in the spring, these systems respond very quickly and the temperature starts rising again. This is a slow-moving stream diagram. Same deal, but even more pronounced. The groundwater is always above freezing. The surface water is always above freezing. So this is the water just right above the bottom. It's not, at the top. There's ice there, but just at the bottom, even though it's very cold there. So what this tells us is that even though it gets really cold up there, that there's water always moving in these systems. And so we went back there the next spring at breakout. So this is, you can see things haven't really started to turn green yet. This is in, I think this is in late May. And when you walk here, there's still ice in the, in the soil. But when we did our fish sampling, fish were there again, no problem. What we found is that what happened with the fish between the summer sampling and the next spring-summer sampling is you had shifts in the age classes. They're basically moving, so the sizes of the fish changed, but where they were. So a little bit on the wetland side, here is moi with my handy knife doing my high-caliber ecology work, which is slash them and bag them and weigh them. So the way we did the wetland work was to, in the initial sampling, was to look at a stream reach and put in a series of transects that were perpendicular to the stream. And in these transects, we measured all the woody vegetation, living trees, standing dead trees, down dead trees, that sort of thing. And then focusing in on the stream, we had smaller plots that we did shrubs. So we measured what was there, how many, sizes, all that sort of thing. And then within the shrub plot, we also had an herb plot because our attention was mostly on the wetland vegetation adjacent to the stream. So that was what we did in the first sampling. When we went back that second summer in spring and summer to look at after the winter activities, we also wanted to know, did it matter where you were on a headwater stream? If you were far up where it was covered with plants completely, or further down where you had a wider stream. So we redid the same sampling regime, but we did it in 3 different locations along the headwater stream to look for uniformity and pattern in the vegetation. Well, the sort of result of all of that analysis was that these are fairly diverse systems. We recorded almost 50 plant species in these plots that were within a meter of the stream. But if you look at the percentages of the plots, so hundreds of plots that we had, that you see that most of them are fairly low, and then here you have a couple of, then you get this big jump with this Calamagrostis. It is like plant that dominates the scene everywhere. So that's the first take-home message about the wetland vegetation, is the importance of Calamagrostis. We did ordinations on this data, and if you just looked at a display of the sites of the plant plots, you don't see much of a pattern there. It's kind of like shotgun. But it turns out that there is a pattern, and that the sites that are over here, these 4 sites, are headwater streams that had their origin in a vast peatland. And when you get in the interior of a vast peatland, it's really low nutrients. And so basically you find a lot of mosses, not a lot of higher vegetation, and it's sort of low stature, not highly productive. These 2 sites are the sites that were the furthest ones up on watershed that were basically in forest. So these 2 end members, you can sort of just, members of the 2 ends, you could describe them, and you had all this stuff in between. But when we looked at the features at the plot scale, the stream reach scale, and the watershed scale, there were a number of variables that helped explain, or they were correlated with these 2 axes. There were lots of stream reach variables that were associated with this distribution. All of these arrows are basically the arrows for going up the slope to the steeper streams, where you have cobble and gravel and sand and all that stuff. They were all related to everything over here. This is related, these arrows are related more to very fine peaty sediments, things associated with these slow, sluggish, moving wetland systems. So we've explained a lot of the variance that we found in that. Published that paper in Wetlands last year. But the key thing is this grass, this blue-joint grass, Calamagrostis canadensis. It's not a plant that is only found in wetlands. It is in the boreal zone across Canada and Alaska and the different species in Asia. It's everywhere. And when you go to the literature and read about Calamagrostis, mostly what you find is that foresters have studied it because they're interested in its competition with seedlings of trees. And basically, how can you control it so that you can have trees grow? But it's also clearly a dominant in these wetland habitats. So in particular, well, let me go back to, yeah. So this first axis is really an environmental gradient. It's the stream, differences between the streams. This we interpreted as a competition gradient driven by how much of that grass, Calamagrostis, was there. So one way to express that is if you look at the relationship between the importance value of Calamagrostis on axis 2, and you see this very nice pattern. These are the 4 sites that are way in the interior of these vast peatlands where there isn't a lot of Calamagrostis. So if you take these out, that's a fairly tight relationship. And if you look at the relationship between the importance value of Calamagrostis and the number of other wetland plant species, you see this pattern, which tells you that it's really controlling the deal. So in some ways, this could be almost equivalent to what is called an ecological engineer. It's a plant that once it's there and it takes over the landscape, it's controlling what's going on from a plant perspective. And so I'm going to focus a little bit more on Calamagrostis. So I take you back to a picture that was almost like one of the early ones where you have a stream that is sort of emerging from a vast stand of Calamagrostis. They had a beetle kill up there about 15 years ago, and a lot of the infilling of the streams is due to old spruce trees falling over and then basically the wetland plants growing out over it and just forming a ladder over the whole thing. Here's an example of what it would look like in the spring, showing you that when the stream gets up, for a brief period of time, it does flood at least the stream area immediately adjacent to it with enough velocity to interact with the Calamagrostis that's there at the creek bank, and particularly the dead Calamagrostis, this stuff. And this is just to point out that these are really dynamic systems. They can change overnight. They'll change for 100 years, and then they'll go back to what they were before, mostly driven by beaver, the other big engineer up there. So let's talk a little bit about Calamagrostis. These are data from the second year where we sampled sites at 3 different locations along the headwater streams. And what this graph shows you is that it really doesn't matter whether it's a high gradient stream or a low gradient stream, that the amount of biomass that is there really doesn't differ that much. None of these things are significantly different from each other. And there isn't really any difference across where you are on the first order stream. The bottom diagram shows you, if you compare these bars, about 500, to these, which are 3 to 500, that the vast majority of that biomass associated with the stream, the wetlands adjacent to the stream, is Calamagrostis. It's the dominant plant that is there. So what happens to it? Now here's one of our sites, 1203 we call it. So there's a stream here. Lots of, you see the Calamagrostis growing on the edge of the stream. So if you're down there looking at it, this is the kind of thing that you'll see. Even in the middle of the summer, once the Calamagrostis gets up to the point where it produces inflorescences, it starts to fall over, they get rains, they get things like that. So this stuff is hanging over the creek bank, and you can actually see it interacting with the stream there. So one of the studies we did was to sample along these two stream types, looking at the creek bank, and then as we went along the creek bank, just recording the frequent, or the cover of Calamagrostis plots as we went along. And the picture kind of tells the whole story, that there's a lot of Calamagrostis there, and you can see that in these data, which is the mean percent cover of low gradient and high gradient streams. So it doesn't matter which stream type you're in, there's a lot of Calamagrostis, and there's a lot of it right at the creek bank. And what happens when that material falls over into the creek is that in the low grade, in the high gradient streams, the water, after the spring flows, the water goes down very quickly. And so you have the water down here about this deep, and you have the Calamagrostis, which has been hanging over the creek bank, and some of it will be in contact with the water, but most of it won't be. When you're in the slow gradient streams, which have deeper water, once that Calamagrostis falls over into the creek, more of it is in contact with the water for longer periods of time. And you can see the difference between, this is probably 2 to 3 years of litter hanging off the creek bank, and you can see this bit is fairly dry, and all this stuff is very wet. As you go down here, you can see it's much wetter. If you could feel that, you could begin to feel you can see algae are on it, fine organic films are on it. So this becomes a substrate that is important to the aquatic food web. So it becomes a carbon source to the stream. It's a site for nutrient enrichment. It turns out that in the high gradient streams, this stuff becomes enriched in metals, copper, boron, things like that are coming in the groundwater, not so much in the low gradient streams. It helps stabilize the stream bank, and as you could see when we were diving down to find the stream, it really can influence the stream microhabitat. So here's an example of that sort of interaction. So the wet part of the litter versus the dry part of the litter becomes enriched in nitrogen. So that becomes a nutrient source for aquatic food webs. And to show sort of the further consequences of that, Becky Shaftel did her master's thesis looking at Calamagrostis. And what she showed is that the concentration of phosphorus and the concentration of nitrogen in litter packs of Calamagrostis that you would put in the stream, the concentrations of those two nutrients went up as the concentration of those two nutrients went up in the stream. Again, fairly obvious there that you're going to increase the concentration, and as a result of that, you're also, if you increase the nutrient concentrations, you're also going to increase these two graphs up here. You're going to increase the decomposition rates. So this is the rate at which this material is being processed and then becoming incorporated into the stream aquatic food webs. So the next part of the story is about alder. I mentioned that as a shrub to small tree. It fixes nitrogen from the atmosphere, so it takes N2, puts it into forms that are biologically active, and then roots decay, leaves fall, they decompose, so it becomes then a source of nitrogen for other organisms to use. This shows a map of the distribution of alder across the Kenai lowlands at the scale of individual headwater streams. And you can see that there are some areas where there's as much as almost 40% of the landscape is covered by alder. And of course, you have some areas that don't have any alder. The distribution of alder across this landscape is not random at all. So if you look at this map, the purple areas are where the alder are. And you can see most of the alder is in areas where you have slopes of about 2 to 10%, so it's not on steep slopes and it's not on really flat areas. And this shows its distribution relative to elevation across the study area, and most of it occurs at these sort of intermediate elevations. So we did some studies, and Becky Shaftel was the lead on these for her master's, where we compared a watershed that had a lot of alder on it to a watershed that did not have a lot of alder. And this is where she did those studies of the decomposition of the litter and things like that. We also measured the nitrogen coming out of these watersheds. And it turns out there's a strong relationship between the amount of alder on the watershed and the concentrations of inorganic nitrogen in the stream water. And if you look at these three graphs, this is May, June, and July, you see a real difference temporally. That in May, which is the time when the water is just, the snow's melting basically, and the ice is melting, that there's a very strong linear relationship there. But by July, that relationship has changed. And into September, it's changed too. So that it's no longer linear, it has a different shape completely. And what this told us is that the groundwater that is coming off of the land after the initial flush of snow is away and ice is away, that nitrogen is coming off of the landscape, and that basically is the source of the nitrogen to the stream ecosystem. So it helped us understand the sort of link between what's going on with the alder and what's going on with the stream chemistry. Another question you might ask is, well, okay, so how is that related to the organisms in the stream? Can you show some direct contact there? So one of our summer samplings, we sampled two streams, one that had, here's alder, one that had a lot of alder, and one that had almost no alder on it. And we went out and we sampled, we scraped algae off the rocks, any other mosses that were in the stream, so those are the primary producers. And then we collected all the invertebrates that we could get, so those are sort of in the intermediate trophic levels. And we collected also organic matter of different forms, large particulate organic matter, leaves, branches, things like that were in the creek, and also fine particulate organic matter and fish. So we tried to get elements of all components of the food web, and then Ryan subjected those to isotopic analysis. And so the sort of the broad view of it is this, that the pink are the areas, everything associated with the watershed that had a lot of alder on it, and the other areas are the watershed that did not have much, had no alder on it. Main point is that they differ. There's overlap, but they basically differ. The nitrogen signal for alder is right here. So it's very low. And you can see the fish also are different in the two systems. They used a mixing model where they looked at carbon and nitrogen and hydrogen and nitrogen, and it allowed them to project how much of a fish's body how much it was getting its nitrogen from the landscape versus how much it was getting from in the stream. So the two terms here are autochthonous, which is material that has its origin in the stream, and allochthonous, which is material that has its origin outside the stream. And the first thing you see from this for a coho and for Dolly Varden is that the data for the autochthonous are very different, except for this one dolly, than the allochthonous. And they use this modeling approach to predict that on average, about 65% of the nitrogen in these fish was coming from outside the stream. And that was very surprising to us because these streams, the pictures I've shown you, you can see they're mostly out in the open. A lot of sunlight coming in, long summers up there. We had thought that most of what was going on in the stream food web was going to be from within the stream. It turns out it's not the case at all. Most of it is coming from outside the stream. So that takes us back to alder. So alder is the primary source of nitrogen to these systems. We go back to this diagram. And now what we're interpreting this to mean is that there's a real seasonal component to alder, the nitrogen getting into the stream from the alder, with much of the late summer, midsummer, and late summer stuff coming from groundwater, that's all that's keeping those streams flowing at that point in the year. They don't have a lot of rain in the middle of summer, so the flow is basically groundwater seeping into the stream from the surrounding watershed. So we did a study last summer where we asked the question of, does it matter where the alder is? If it's far up on the watershed versus close to the stream, does that have any consequences to the wetlands adjacent to the stream and ultimately to the fish? And my thinking on that was that if the alder is close to the stream, there's a greater chance that any nitrogen leaves that system, the alder system, is going to make its way to the wetland and then to the stream, whereas if it's far away, there's a greater chance that it's going to be taken up by vegetation on its way down to the stream. So we looked at the position of alder, and the way we did that was Mark Range's group was working on this with me. We set up transects of groundwater wells. So we had alder up here, and then we had a series of wells that went down to the wetland just immediately above the creek. And we replicated that in places where we did not have any alder up on the watershed. And what I did is I studied the wetland right here, and I fertilized plots. And if alder was, if nitrogen was making its way down to the wetland from the adjacent alder, if it was happening all the time, then by giving the vegetation some nitrogen, you would get a response, but it wouldn't be nearly as great as a response if the alder was far away and the nitrogen was all being taken up before it got to the wetland. So basically using the plants as a phytometer to tell me whether alder position had any difference on productivity of the Calamagrostis. And while I was doing that, Mark and his students were looking at groundwater and surface water that was flowing down here, and also stream water. And we did this study based on flow path analysis, where we looked at watersheds and we looked at where the alder was, and you can use flow path analysis. You look at the topography of the land to tell you where water is likely to flow. So we set this experiment up in places where we knew water was coming down from up there. Here's such an example. This is one of our sites with alder near. There's a big patch of alder. The slope of this, you really don't get it from this picture. It's about like that. And you can actually see little streams flowing down here. So this is all within the flow path from that site, and they're setting up the fertilization experiment in the spring. You might ask, well, why did you set that up? Do you have any idea why we set that up? Because our plots are little teeny PVC pipes. Why that?
Might grow up too much, you miss it.
Yeah, you come back here in July and you can't even see the place. And you're walking, little guy like me, you know, I'm up to here, or here in vegetation. So to summarize that, so this is the result of the experiment where we fertilized and didn't fertilize. We compared alder being far away, alder being near, and a control site where there was basically no alder. And the prediction was that we would get a big response in the control site where we didn't have any alder. We would get a response not quite as big at the site where the alder was far away, and we get less of a response where the alder was close. And you can see that's exactly the way it worked out. Looking at the water side of that experiment, what this shows is that if you have alder near or alder far, that in the groundwater you're going to have more nitrogen than you would have in a place, in a watershed where you don't have any nitrogen, so that makes sense. The hill slope surface water, this is water where they could basically walk down that flow path. They saw surface water flowing. They just put their sampler there and took a sample, sampled that water, and this sort of fits with our model too, that if you're close to where the alder is, the concentration of any surface water that's flowing is going to be higher in nitrogen than if the alder is far away compared to where control. And then the other, this is looking at the water down in the stream, and that's sort of at a watershed scale, but this is more a measure of how much alder is on the watershed than it is kind of position of the alder. And here's another way of looking at that. So here are the symbols are surface water, groundwater, and stream surface water. So here's the distance from the alder patch going down toward the stream, and you can see that when you have alder present, that the concentration is high, very close to where the alder is. And it drops off rather quickly. So something is taking up that nitrogen that was coming out of the alder patch, and it keeps dropping until you get almost to the stream again. You get basically to the wetland that is immediately adjacent to the stream, and then the nitrogen kicks back up. It turns out that you have the alder patch, and when there's enough water, there's surface water flowing down this flow path. A lot of it's becoming deeper groundwater and eventually emerging in the stream, maybe a mile downstream, but it's emerging in the stream. But the other thing you find in almost every situation there is where the slope ends and the wetland begins, you find seeps. So these are areas where very shallow surface water or very shallow groundwater is emerging. In some cases you find where even deeper groundwater is emerging. This is an example where deeper groundwater is emerging almost immediately adjacent to a stream. This is like going to Yellowstone and finding where water's coming out of the ground. Only in this case it's ice cold and clear and not hot. This is more typical, where here's the break-in slope, here's the wetland, and you find this very shallow groundwater emerging. You can see it colors the leaves of the vegetation. Here's another example where there's enough water coming out here that's wet enough that you actually have a very different plant community in that particular location. And here's another one where it's emerging out of a shallow slope right here. So what have we learned from these various components of the study so far, which is still ongoing? So getting near the end here. It turns out that our original thinking that the geomorphic setting was going to be a key driver in the ecology of these headwater streams is not the case at all. So it's much less important than other features of the landscape, particularly the slope of the landscape, and then a number of factors that we sort of lumped together in site wetness, basically. These slow-moving systems are just wetter than the ones that are on these gradients. So that's one important thing. I hope I've demonstrated that this one plant is really a key species in the ecosystem, key wetland species. Whether you would elevate it to the category of ecological engineer, we don't know yet. I probably wouldn't do to go that far. But it's clearly important because what it does is it influences the diversity of all the other wetland species associated with the headwater streams. And perhaps even more importantly is we're demonstrating, and through our studies and ongoing project, part of the ongoing work, that it is really linked to the aquatic food webs. So it's just not a wetland plant there that's influencing other vegetation. It has a key role in the characteristics of the stream ecosystem and moving its way up to the fish. We know that alder is important. To give you a little sociological background information, many people up there view alder as a trash plant. If you're a landowner, you might even want to get rid of it because it's not particularly good for burning for wood. It kind of obstructs your view. It forms these dense patches. So, given free will and free spirit of Alaska, often landowners will just get rid of it. So what we're sort of developing here is a story of, well, you might want to think twice about your alder on your land. There may be other reasons for thinking it as being important. The other thing that we haven't totally nailed down yet is that from Mark's water chemistry work and from this recent project looking at flow paths, it seems like Adler is important for two things. One is on a regional scale, i.e. the whole watershed, the amount of Adler there is really key in determining how much nitrogen is getting into the stream system through primarily groundwater flows through much of the growing season. But that the position of Adler relative to the stream in these flow paths, that may also be important because from the take-home message for me from that last project we did is that if you have Adler sort of immediately upslope from the wetland that is adjacent to the stream, that wetland potentially could be a hot spot. That could be a place of high nutrients, high biological activity, compared to the place just immediately downstream that is not connected to a surface flow path or a shallow groundwater flow path. So there may be different ways that Adler is linked into the stream network. And then, which I just mentioned, that these local flow paths and the presence or absence of Adler may be a key factor in controlling the productivity of Bluejoint and the linkages between that and the streams. Now, the last thing I want to talk about is, I mentioned at the beginning that they only had mapped the streams up to where the adults go, and they hadn't looked at much beyond that. Well, it turns out now, because we have actual data of fish in streams from probably 75 sites now, they now have changed their maps. So those headwater streams are now listed as salmon habitat for the management community. So that we think that's important. And if the model that we've developed for the lower Kenai can be extrapolated to other parts of the wetland, we think that from a regional context, this potentially is going to be important because it'll allow them to expand basically the network of sites that are really key to salmon production. So we think that eventually is an important component of what we're doing. Now, going to the last couple of slides, so what we feel is that landscape integrity and management of upland habitats so that people don't make the wrong decision, that is really going to be an important component of determining how successful over the long term these headwater streams will be in supporting juvenile salmon populations. And it's only if you adequately support juvenile salmon populations will you support things like this. And what we think the message is that these big dudes are clearly tied back to the landscape and to the wetland vegetation. Now, how do you tell people, what your message is? And so this gets at something I think ecologists do a very crummy job of because we're not trained, we don't know it, which is we think eventually we understand how these systems work. How do we communicate that information to managers, to economists, to people that really drive the train? And so that's what we're trying to do. So we have this sort of map of the wetlands of the Kenai lowlands. We have the map of the landforms in the Kenai lowlands. Now what we have is for categories of fish, and I'm just showing you one of them, the large coho. We now can generate maps of the Kenai lowlands that show for every headwater stream on the Kenai lowlands what the probability of that stream is as a very good habitat for that fish, very high probability they're going to be there in good numbers, to a very low probability and an intermediate. And we tested this model last summer. What we did is we built the model, which is driven primarily by slope of the land, distance from the ocean, and the size of the catchment. In this case, distance from the ocean, these large fish migrate from this side of the landscape over to that side of the landscape. They basically move. We're trying to nail that down now. So we have predictors, and what we did is when we did the first generation of these predictors, we then randomly picked sites that we had never been to and went out there with our fish sampling equipment and sampled the fish to see whether our predictions were good. And it turns out they were quite good, and it even helped us refine these models even more. So we think we have something that's very robust. What we need to strive to do next is to figure out ways to link all of this information on these headwater streams in ways that will ultimately lead to them making suggestions for what people ought to do on the land. How close you should come to a stream, stay away from flow paths that connect alder to streams. We think we know, what some of the messages are, but we have to figure out a way working with other people to get that message across. So with that, I'll leave you with that picture and some of the folks that I haven't acknowledged so far. This is Phil North, who was a visionary guy at EPA who saw the importance of this work and gave us money. So we like Phil. And these are the people from the Kachemak Bay Research Reserve that are, make work up there for us. We had a summer intern. The Estuarine Reserve Program has summer internships, and so every summer we get field hands to help us. And Kristen came up one summer and fell in love with it, and went back. She was going to do a master's at Rochester Institute of Technology. So in our most recent grant, we included funding for her to do her master's, and she's doing some really interesting stuff where we have these solar-driven sampling stations on streams, and then we catch fish and put little tags in them, and when they go past that point, there's a recorder that tells you each fish is identified by its own number, so we can tell when the fish passes that area. So we're trying to get information on fish movement within the streams. So she helped us a lot with that, and that's going to be her master's thesis. The other thing that she'll be helping us on with next summer, I hope, is that we're at the point now where we think, because of this alder connection, that the streams which have a lot of alder associated with them, we think what's limiting productivity there is carbon. And the streams which don't have much alder, they're being limited by nitrogen. So next summer we're going to do a pulsing experiment to test those hypotheses of what's limiting productivity and subsequently fish growth rates in the stream. Then they have a series of volunteers and grad students, and then there's a technician in my lab who grinds all the calamagrostis when it comes back. So thank you very much, and if you have questions, I'll be happy to answer them.